everyone can see my screen to the Journal of Physiology Virtual Journal Club. The theme today is going to be lifelong endurance exercise, improve slow but not fast twitch skeletal muscle fiber. And I'm going to be your host for today. So before we start discussing the paper and talking with the panelists, I just want to introduce me and introduce the panelists just briefly. So my name is Vini Mariani. I'm a first year PhD student in exercise physiology at the University of Florida. We have Dr. Grosicki. He's the primary author in the paper that we're going to discuss. He's from Georgia Southern University. We also have Dr. Scott Trappe. He's the principal investigator in this paper, and he's from Ball State University. And we also have Dr. Russ Happel. He's the editor. He's an editor in the Journal of Physiology, and he reviewed this paper. So he's gonna share his perspective as well today. And he's also from University of Florida. So just briefly, I want to mention that you also can be involved in the journal by hosting a future meeting as this one. You can also write a journal club article on a recently published article, and also can submit your research. And if you have any questions, you can email the email addresses here that appear in your screen right now. So the paper that we're going to discuss is the single muscle fiber contractile characteristics with lifelong endurance exercise. So the first step, I'm going to just mention why I chose this article. Then I'm going to pass to Dr. Grosicki and Dr. Trappe to discuss a little bit more about the rationale, methods, and findings in this paper. And then Dr. Happel will give his perspective, what made this paper publishable. And then we're going to have a Q&A. So on your screen in the bottom, you can see there's a Q&A function, and you can use that to send your questions. You can do that anonymously, but also would be nice if you can share your name and institution. It's always nice to see familiar, in this case, names, not faces, but names. And then afterwards, we will have a networking session that will take approximately 30 minutes. And we encourage you to participate. It will be a lot of fun, and is another way to interact with the panelists. And we're gonna share the link. I think we did it right actually in the chat for the networking session. So as soon as the Q&A is done, you can move on to the breakout rooms. So before I pass to Dr. Grosicki and Dr. Trappe, the reason why I chose this article, first of all, just to give a little perspective, about 28% of Europeans and 23% of North Americans will be 65 older than 65 years of age by the year 2050. So it's important to understand what's happening with our physiology as we age. And we know that there's a muscle atrophy and dysfunction associated with the aging process itself. But what I found that is complicating aging research is try to differentiate aging-induced and inactivity-induced muscle dysfunction, because as we age, we tend to exercise less. So it's difficult to separate both. But what I really appreciate in this paper, I think the authors did a fantastic job in the recruiting, in which they recruited young endurance exercisers. And for their control, the older population were physically active and didn't have comorbidities for the most part. And they were always also able to recruit master athletes, so lifelong exercisers that are doing the exercise with the goal of performance, but also they also recruited lifelong endurance exercisers that are training for physical fitness. So I think it was an excellent recruitment, and this way you can kind of eliminate the inactivity factor of aging and focus in the role of exercise. I think that was phenomenal. Also, because one of my interests is biophysics, I really enjoy the use of isotonic contractions to measure muscle function at the cellular level. I think gives a better perspective of overall function, because you can estimate power from it. And typically in single fiber studies, we use PCA force curve to try to get measurement outcomes of calcium sensitivity, isometric force, but sometimes that doesn't give us the whole holistic view of muscle function. So using this isotonic contraction, I think is an excellent way to investigate muscle function at the cellular level. And also, this paper gives evidence at the cellular level that other exercise mobilities, such as resistance training, should be performed as we age to specifically preserve fast twitch fiber function. So now I will pass to Dr. Grosicki and Dr. Trappe to discuss a little bit more about the paper itself.
Yeah, thank you for the great introduction there, Vini. And you picked up and hit upon a number of points that I think were also, really important to us when we worked on this investigation. To give you a little bit of a background about myself, I did my master's at Wake Forest and had the unique opportunity to serve as an interventionist on the life study when I was there. And so that was really a fantastic experience, and particularly getting my hand and getting a role in working with older adults with more clinical morbidities and impairments. And when I began talking to Dr. Trappe, and when I reached out to him, he kind of presented basically the whole idea behind this life study to me. And it was one of the things I think that really solidified my decision to pursue my doctoral training at Ball State, was really just this, what I found to be genius idea that they had. And so many people, when we're studying aging and how to promote healthy aging, tend to focus on the older adults who do have these clinical impairments, which is great, and it does provide us with that perspective. But I think what was really so novel about this investigation and the lifelong exercise study in general was that we comprehensively examined aging physiology from the perspective of extremely, probably the most successful agers. And really, I do feel as though it's important that we have that well-rounded picture of what aging can look like from both the people who have the worst clinical outcomes and the people who are not aging as successfully. But then we can use these successful agers as this really unique and attractive model to understand how we can curtail or maybe even totally offset some of the traditional age-related deficits that we see. And with that, I do think it's probably relevant that I turn it over to Dr. Trappe here, who obviously was a wonderful mentor, and it was a great opportunity to work on the Human Performance Lab research team during my time there, because really he'll be the one to provide probably the most insightful perspective as to the conception of the whole R01 funding mechanism and grant that actually funded this project.
Sure. Thanks, Greg. Thanks, Vini. Welcome, everybody. This indeed was a fun venture for us to get into. I guess, honestly, it was sparked a bit by just our own personal interests with backgrounds as exercisers and athletes. But, early in my career, we started studying aging, in particular, non-exercisers that were in their 70s and into their 80s. Bringing them into the lab, training them, resistance training them 3 days a week. And we reversed some of the classic clinical, we'd see about 7% hypertrophy of the thigh muscle. And at that point, we had just got our single fiber lab up and running around the mid 90s. So we were probing into the slow and fast twitch fiber adaptations. It kind of was being pretty well established by that stage that fast fibers atrophied more, there were less of them. And so we were looking like how resistance training would target that. Really found over, a series of studies that we're doing in the lab and other people were doing that the older people did quite well with the resistance exercise, but it was primarily a slow twitch type response, even in the older people. Yet when we did the same protocol in younger people that were non-exercisers, it was a fast twitch response. So there was this sort of divergent cellular approach to the adaptation. So the idea behind this was what about people that have used their fast twitch fibers their whole life? So we set out to begin to recruit some lifelong exercisers. We knew they were there just from our own personal interactions. But also felt like we were at a bit of a critical mass. The timing here, I think, was important. These people that we went out to recruit ultimately were part of what we've called the exercise boom when it took off in the late 60s and early 70s. These people are now into their 70s of age and have been engaged in exercise as a hobby, as a lifestyle, their whole life. Really the first generation, in my view, that's done that, because prior to that, it was more of a work type mentality, for people to be active. And society began to engineer that out. So, the exercise mantra, particularly with running, took off in lifestyle. So that was the idea. And then to, get under the hood with, looking at whole body responses from the cardiovascular system, maximal heart rate, getting into the, muscle from a whole muscle perspective with MRIs to look at what was happening, to our muscle biopsy work, to look at enzymes and look at a single fiber approach, which we're really interested in, particularly the fast fibers. And that's what this paper and Greg's dissertation focused on, and also some molecular work that's still ongoing to try to understand these things. So kind of a whole body to gene approach with the study. So that was the thinking. And then we had a lot of energetic people in the lab, and Greg and others, because it's a team of people. I think the people have done human research know to bring these people on board. And I'll just say anecdotally, working with these people were awesome. I mean, bringing them into the lab, they were just great. And I would say actually inspirational. It made me want to exercise even more. So I was okay, I want to be them. They were just great. They retired and they had extra time on their hands, so they doubled down. If you look at the paper, it's interesting. Like a lot of times people say, we broke it down by decade. Like these people started putting more time into their exercise, and it's true, because what happened was their life, their kids went off to college, and when they had more time, their hobby was exercise, so they doubled down and got even more involved with the exercise. And they had great logs and things of that nature. So it was wonderful interfacing with these people. So the data is reflective of that, I guess. So I'll stop there.
Thank you, Dr. Trappe and Dr. Grosicki. Now I'm going to pass to Dr. Happel to give a little bit of his perspective of the paper and what made this paper publishable. I think you're muted.
Here we go. Thanks, Vini. I'm unmuted now, right? Good. Okay. As a reviewing editor, our job is kind of multifaceted. I don't know how many people know kind of what happens behind the scenes, but I'll get contacted by one of the senior editors and say, okay, here's a paper that we think is within your expertise. Would you be willing to serve as reviewing editor? And basically, my job then is to do an initial screen of the manuscript, make sure that the work is novel, the question is scientifically significant. And that generally speaking, it looks like the methods are robust and that the findings advance understanding. So if it kind of passes that initial checkbox, then we advance to selecting reviewers. So, and then after that point, reviews come back in some weeks later, and hopefully there's unanimity in the reviewers and makes my job easy. Sometimes there isn't unanimity and it's a little more challenging, and you then have to weigh in a little bit more. So in the context of this particular manuscript, like Dr. Trappe, I have a longstanding interest in aging muscle biology, lifelong athlete as well. We've studied some of these people ourselves in the lab. And so, when I see this paper come across my desk, I immediately go, yes, I would like to be the reviewing editor. And then, perusing it, it's like, yes, okay, awesome. This looks really good. As many of you will know, Dr. Trappe's group has got a long history in this area. They've made a lot of really important contributions to this. And so seeing them put another chapter in this book, I think is really exciting when you get an opportunity to see it. So I was very enthusiastic. It checked all the boxes for me. And thankfully there was a lot of agreement in the high caliber of the work from both reviewers. So it made my job very easy.
Thank you so much, Dr. Happel. So now we're gonna open to the Q&A. And I think just to icebreaker. I'm gonna start with a question, if you don't mind. One in one part of the discussion, you compare the results from the paper that you put out in 2019 that you use women, and then in one table of in the paper you compare the results from that paper with this one, comparing men and women, and there's a couple differences. So I was wondering, what do you think is the main cause for that difference between men and women? Men and women, do you think is the estrogen levels that could be affecting the way that the source of this discrepancy between the genders?
Go for it, Greg.
All right. Yeah, great question, Vinny. That was one that we certainly kind of something that was one of the unique things about this paper is that Kevin Gries had published his paper in 2019, and it was kind of nice to have that comparison to the women data. And so, yeah, I think hormones are certainly one possible contributor to these sex-specific differences. I think there are other things, a little bit of cultural influence, things of that nature. The women in the study, whereas the men, one thing we were thinking about, the men, most of them were literally able to train consistently for 50 years. The women also, many of them had children and things like that, so had to take time off from their training for maybe more extended periods of time. So there are certainly cultural differences as well between the men and the women. Admittedly, we did study a few less of the women than the men. And so another thing that you'll see is that for the men, we were able to even subdivide them by basically competitive status. Those who were kind of still competing in various races, and there were, as Dr. Trappe alluded to, there were some just phenomenal people to work with. A lot of them, particularly in this lifelong exercise performance group, felt as though even though we were there studying them, that they still had to be auditioned. So we're sitting with them in the biopsy room and they're telling us all about their races. And it's like, I hope I'm eligible for the study. We're like, trust me, you're eligible. Like, they don't make movies about your performance. And we don't want to study you. So it was just incredible. And the women were equally as impressive, but because we didn't have the same number, we weren't really able to parse out the possible contribution of how competitively were they doing it. Were they doing it more for competition, or were they doing it more for fitness related purposes? And so one of the things we did see with the men, and this was more specific to the type 1 fibers, which is actually where we saw the greatest number of sex specific differences, was that there was appeared to be, particularly with power, underlying differences in force and velocity that contributed. And so another possible explanation for this is simply that, there weren't as many women in the study, and so we weren't able to really look at how intensely they were going at their training throughout the lifespan. That's a great question, though.
Yeah, that's a good summary, Greg. I think I'd just add, I think it's clear, you get the literature now, and there's a lot of data showing there's sex specific differences here, and in the biology. I think it's a complex issue when you start thinking about the coordination and communication between all these organs and stuff that's probably going on as they get older. And as Greg alluded to, it's interesting because when the exercise boom took off, it really was a male dominated boom to start, but then you had a few mavericks that broke through at the Boston Marathon, Title IX in 72, and so that was just a bit delayed. So there just wasn't quite as many as the numbers out there. And we had a couple women that were really competitive at high level, going to track meets and setting records and things of that nature. But there was also a little bit of a mentality difference. The guys that were in the competitive, they weren't a whole lot different than the 20-year-old competitive guys in terms of their attitude, right? I mean, it was kind of interesting. Even competitive with each other, and want to know how everybody else was doing and to size themselves up and stuff. So it was good. I don't think we can't answer that question, but hopefully in the future, not only our team but others will continue to pursue this, because I think it's important. On one side you might look at there's the men did better in some respects at the cell level, but it's hard to argue with all of that stuff. You look at the IMAT data that was in a paper, a companion paper from this stuff, the men tended to do a little better than women, but by and large, women outlive the men in society. And I think both these groups of men and women are doing quite well, independent living, very active. So, probing into some of these details that would parse out, I think is the technology and tools are starting to come on board where I think we'll learn a lot as we go forward.
Thank you so much. As we wait for more questions in the Q&A, I have another question. I can prepare. I thought it was very interesting, the discussion that you mentioned that perhaps there is a muscle-specific adaptation. So perhaps if you would get a biopsy from the gastrocnemius or soldiers, you could see something different. Perhaps due to the biomechanics of running and cyclists, since we're dealing with two different types of exercise, right? In different range of motion in the knee and so on. Do you think that that you would see a difference if you, for some reason, you decided to get a biopsy from the gastroc or the soldiers? Would you expect to see something different results, and what would be the results? Let's say where we would likely to see the difference.
Yeah, that's another good question, and actually a question, Vini, that one of the reviewers asked us. So kudos to you for asking that question. Very insightful. We certainly know that there's muscle-specific differences in adaptation to various exercise training perturbations. Nick Luden showed that almost a decade ago in marathon runners, comparing gastroc and soleus and VL. We've seen it with space flight, and now, we expect we would see it here. And so certainly one of the limitations of this study was that all the biopsies were acquired from the VL. Now, I think that that's also probably one of the strengths, because this might be one of the more functionally relevant muscle groups. It's certainly one of the ones that the crew at Ball State has studied the most, and probably has the greatest implications for generating whole muscle power of the lower extremity. So looking at falls, which is one of the things we were particularly keen on. Another advantage to the VL, as we know, is that it's quite a mixed muscle with a pretty even distribution in just normal healthy individuals of slow and fast twitch. So by studying the vastus lateralis, we were able to really gain perspective as to the effects of the lifelong exercise on both slow and fast muscle fibers. So I certainly think if we were to do it again, we would choose that. But to your point, certainly there would be differences. We know the soleus, for example, is primarily type 1 fibers. All of the subjects in this particular study were engaging in at least regular forms of physical activity. We talked about it in this paper, and we talked about it more in some of the other papers about how even the old healthy cohort was taking 5,000 or 6,000 steps a day. So they were up and moving around. And really, it's just postural. It's a postural muscle, the soleus, mostly type 1 fibers. And so we might not see such a disparity in terms of the contractile mechanics in the single muscle fibers of the soleus, simply because it's being used just for regular lifestyle activities. Now, I think to contrast that with the muscle of the upper extremities, I think, we would have had totally different findings. All of the individuals in this study were lifelong aerobic exercisers, and while some of them may have done resistance exercise from time to time, it certainly wasn't really any of their focus. And so I doubt we would have seen nearly the same benefits we saw, particularly in the slow muscle fibers, for muscle fibers of the upper extremity. And there's data, neuromuscular data from great data looking at motor unit number estimates from Jeffrey Power, I believe when he was doing his postdoc, showing basically exactly that in these masters, pretty elite masters runners, seem to preserve motor unit number in the lower extremity, but not the upper extremity. And so, as we allude to in the discussion, and with what we've seen from the fast muscle fibers, there's certainly need to study other muscle groups, as well as looking at interventions to preserve the muscles of the upper extremity.
Yeah, I think it's a great summary, Greg. Well, I guess just to add, I would expect differences. I mean, and that's based upon the work we've done with NASA over the years, where we did a multiple muscle approach to most of the bed rest and flight studies and things of that nature, and they just adapt differently. You know, the soleus is a postural muscle, so it's gonna, you know, just the weight bearing nature of it, it's probably almost always trained. But I think what Greg said, though, the VL is a great muscle. That's the most comparative in the literature. You got the mixed muscle, and frankly, it's the safest, right? I mean, when we've done soleus muscle, and we've done quite a few of those at Ball State, I would think twice before I got into it with the aging cohort. It's not as outlined. It's a smaller space to get in and do it. The athletes is a little bit easier to see it. You get into the non-athletes, you got the MRI scan, but it's a little trickier business in my route. Probably why you don't see a lot of that in the literature in older people and stuff, right? So some of that I think comes down to a logistics issue as well. But I do think probing some of these other muscles would be of interest, because I think there'll be, you know, beyond what we're doing. I think you'd want to have some nice a variety of science platforms, so you could really look at, if you're going to make that go at these different muscles, maximize the science return.
Thank you, Dr. Grosicki and Dr. Trappe. Dr. Happel, do you have a question or an insight?
Well, actually, I just want to add to the discussion here when we're talking about upper body versus lower body. And there is this long-standing, you know, inference that this relates to the fact that, you know, we don't, unless you're a circus performer, you don't walk on your hands, right? So we walk on our legs, and you know, that's the reason, because as we're getting older, we're being more sedentary. That's why the lower body has bigger effects, bigger decrements. But I want to caution everybody, we see the same thing in quadrupeds. So you look in mouse models or rat models of aging, the upper body muscles are less affected than the lower body. So this can't be something that's exclusively due to the degree of use versus disuse as we age. And there's data suggesting that it actually has to do with the origins of the innervating motor unit pools, and that there are a handful, there's not a large number, there's a handful of muscles that are more resilient towards the effects of aging. And one of the fascinating things to me is, at least in rodent models, the same muscles that are resilient, more resilient to aging, are also resilient to certain neuromuscular diseases like ALS and spinal muscular atrophies. So there's something about the motor unit pools that seems to confer vulnerability versus maybe, you know, better ability to withstand the forces of aging. So just add that to the mix.
Thank you, Dr. Happel. We have some questions here in Q&A. The first one is from Chris Sandberg at Marquette University. He says, excellent study, Dr. Grosicki and Dr. Trappe. Exciting to see this work coming out in publication. Could you please expand upon whether you think the inability of the lifelong exercise to preserve the size and contractile function of the fast fibers is due to an inadequate stimulus and full recruitment of the fast fibers, or blunted plasticity that occurs in the fast fiber type with aging?
Yeah, thanks for the great question, Chris. And I think the answer to your question is probably yes and yes. There is still ongoing investigation on some of the data, particularly from this study. One of the unique things we did with the study protocol was we did a biopsy just at rest, and then we had these subjects perform a resistance exercise training bout, and then took biopsies following that bout, and then 4 hours after that bout to actually probe for molecular and protein responses to that resistance training. And so one of the purposes of that, including that in the study methodology, was to actually look for basically stimulation and then changes in the molecular and protein levels in the fast muscle fibers, as well as to compare them between the old healthy, or among the old healthy, the young exercisers and the lifelong exercisers. And so I believe, work that should come out of the the HPL in the next year or so, or hopefully will speak to some of those findings, and that should be some really exciting data. We do know there's certainly data to suggest, there's a paper, I believe, in 2017 that showed even at 75% of VO2 max aerobic exercise certainly activates the fast muscle fibers. I guess the question is certainly, I guess it turns more to, is that activation of the fast muscle fibers sufficient to preserve them with aging? And in the context of this paper, or these cross-sectional data, it would certainly seem that maybe it's not quite adequate. And so I think that really speaks to the need to examine lifelong resistance exercisers, which is certainly something that would be super exciting to do. But I do think we need to take it back. When we do a study like this from the whole muscle down to the gene, I think sometimes the further you get away from the whole muscle, sometimes maybe you lose perspective of the bigger picture. And that's that if we're looking at some of these 70, that most of them were about 75 years of age individuals in society, and we ask ourselves, are these individuals successful agers? Are we worried about them from a physical function standpoint? Are they able to ambulate? Are they able to perform their activities of daily living? I think it really begs the question, how important are these fast muscle fibers? We know we make it out as being super important, but these are definitely some of the most highly successful agers. I'm not really worried about any of these individuals having a fall. So yes, I do believe that's not to diminish the importance of the fast muscle fibers, and certainly cells from a grant perspective, right? But these individuals are definitely successful agers, and so I think it really begs the question, if we have these adaptations, these compensatory adaptations we've seen in the slow muscle fibers, is that sufficient? We need to maybe longitudinally study these individuals, and so hopefully that's something that I know it was talked about during my time there. Maybe bring these individuals back in every decade and see how they're aging.
Good comments, Greg. I will add just sort of my own personal curiosity, a couple comments. I think at the end of the day, going into the project and the data I've looked at, the literature, was it an inadequate stimulus or full recruitment? I think, if fiber's recruited or it's not, there's some submaximal data. Really, I think the data, we don't have it specifically with these individuals, but I think the activities that we're doing, there's no question they were recruiting these fibers off and on over time. And it's interesting, at least from the data presented in this paper. So if you look at the size, strength, speed, power profile, the fast fibers, you sort of put it out there. There's some differences here and there, but by and large, they stack up. They look like, oh, there's no impact. You get into Agard's work. It was a while back. They had some resistance training people. I think they published that in MedSci 07. There was a couple other papers suggesting that, RE, in fact, preserve the size of these or do a little better job. I suspect beyond the contractile properties, you get into other aspects of these fibers. It's fair to say, I think, that the endurance exercise did not promote a growth component for the fast fibers in these people like it did with the slow, for whatever reason. But metabolically, I think they're probably completely different. We don't have that data in here. We're working on it. So these fast fibers between the old and young probably do have some big differences that just don't come out in the contractile property profile, where they've probably been sort of all these years suited up to behave a little bit more to support the metabolic activities than they have a growth activity. It's a little counterintuitive that these fast fibers, as a result, they got so big, you'd think maybe that would be more of the slow fibers as well. But nonetheless, I think at the end of the day, there's just some things here that you can't prevent, right? We're slowing aging, we're changing the paradigm. The whole idea here, this is sort of the best case scenario. What are the potential benefits of it? And I thought for sure we would see some benefits here at the contractile property level, and for the most part, they were pretty comparable to the comparator group. Now, those group are pretty healthy too and active to qualify with the inclusion criteria these days. I think if you got some more of nursing home type people, but then we don't recruit those types of people. They got tons of comorbidities. They're on tons of different medications these days. So even the healthy group that were non-exercisers were kind of elite by today's standards. And you look at the step counts of the people in the paper and stuff. So that I think we got to put that in context a little bit too as we interpret the results that are non-exercisers. We're still a great group of people, both groups successful agers.
Thank you, Dr. Trappe and Dr. Grosicki. We have a second question from Kevin Murat. Given the results, how important do you think those fast twitch fibers are in the context of aging? I think Dr. Grosicki alluded that in the previous answer. And a follow-up question, have you probed for markers of denervation in the tissues from these athletes?
I would just say one comment. I think working with the athletes, they're bummed they're losing their explosive speed for competition reasons. So maybe from a big aging picture, they're doing okay, but they were all complaining about slowing down as they got older. So I think they would definitely take some fast fiber benefit. And we are doing some follow-up stuff, Kevin, on this. In fact, you're better at some of this, so I may have to reach out and make sure we're getting all the right assays done.
Yeah, I think Scott makes important points there for sure. And I did kind of talk about this question in the previous question. I think just to kind of talk a little bit more about, the real lack of fast fiber benefit. If we take a look at some of the data that's maybe a little bit more well-known, some of the stuff we've known for decades, specifically if we look at changes in even enzyme concentration, right, and skeletal muscle with aerobic and resistance training. We know we have someone do a whole lot of aerobic training, they have an increase in a lot of these aerobic enzymes, and actually the anaerobic enzymes we know actually go down a little bit. And so really, perhaps what we're seeing here is really the same phenomenon. These individuals have really, in order to optimize their slow twitch muscle fibers and really optimize the proteins, the contractile function of their slow twitch fibers, maybe, for all intensive purposes, their physiology kind of put those fibers on the back burner. And that might be why we're seeing some of these changes. Now, that's not to belittle the importance in the fast fiber and pretty much the, average healthy ager who hasn't been participating in 50 years of lifelong exercise. And them, I think, probably significantly more important. And then the question about, the denervation is great. And I actually have had some recent conversations with Dr. Sundberg at Marquette, who's trying to perfect those assays right now. And hopefully be able to actually do those assays on fibers that have been studied for contractile function. So I'm sure some great insights coming from his lab as well as Dr. Trappe's lab to that question.
Thank you for the answer. We have another question here from Chris Vandenberg again. Yes, along these similar lines, did you have subsets of older subjects that primarily cycle and others who primarily participate in running for their exercise modality? And I think you did. But if so, did you observe any differences between these subsets in the size and the contractile function of these low and fast fibers?
That's a good question. I'll start off with that one. Chris, there just wasn't a clear info. I mean, the reality is most of these people started off running with the exercise boom. That was the thing, right? But that wasn't sustainable over the long haul. They recognized that. They began to switch modes or cross-train. So I don't think we had anybody in the study. We had a couple that were pure cyclists. Had a handful of people that really incorporated cycling to extend their running career. So as we got into the older brackets, and myself, I do this, most everybody starts to mix and match versus they're just pure one sport. type athletes. So there really wasn't enough data to break that out to say, here's what a running phenotype would look like, or here's what a cycling phenotype would look like. And I'm not sure that would be sustainable to get. You might find a handful of individuals globally that would, I've just only cycled my whole life. You're gonna have a hard time finding globally people just run their whole life, because orthopedically they just begin to fall apart. It's not the physiology. They're forced to find other modes of exercise to stay active. And that was certainly the case for the people that we had enrolled in our study.
Thank you very much for that. I have a question from, I think it's figure 4 in the paper, in which you see that the older individuals, the old control, have a greater power, peak power, the absolute power, compared to the young individuals, and even with the the lifelong endurance athletes and exercisers. And then when normalize the power, then those differences kind of change a little bit, but it's still the older with fast fibers, with fast fibers. But then when you normalize the power, the older individuals that are not lifelong exercise still have a greater power compared with both groups. Why you think that? Why you believe is that the source of that result?
That's a great question, Vinny. And one of the things that kind of was the first project I got to participate on during my doctoral studies at Ball State. And in that particular study, which was a subset of participants from the Healthy Aging and Body Composition study, we studied, it was about 5 individuals who were about 90 years of age. So really the oldest old here. And we looked at vastus lateralis muscle biopsies and studied their single muscle fiber contractile function. And when we were comparing, we were really focused on that normalized power, because this presents a real integrated, comprehensive picture of contractile function of these fibers, incorporating power, which we know is probably most important for whole muscle power, and thus the prevention of falls in older adults, as well as normalized to the size of the cell. And so when we were looking at the normalized power of these 5 approximately 90-year-old individuals, I was just kind of comparing it to other data sets from the lab. And what was really fascinating, we started out by looking at some studies in young 20-year-old runners. And then we looked at some 40-year-old individuals, and we saw no real change in the normalized power of the muscle fibers. And we thought, well, this is interesting, but it's not totally unprecedented, particularly if those 40-year-olds have been pretty active, and kind of mirrors what we would see at the whole muscle in terms of size and function. But then we started looking at some of the older cohorts, some of the individuals Dr. Trappe studied back in 2003 in one of his big J physiology papers. And then we compared those to some single fiber data from Dr. Raue during her doctoral studies at Ball State, and then Dr. Slivka, which came a couple of years before me. And what we saw was really this stepwise increase in normalized power of the type 2 muscle fibers as these individuals got older. And actually we saw the greatest normalized power that had really ever been reported in the human single muscle fiber literature in the type 2 fibers of these 90-year-old individuals, which we thought was just remarkable and totally dogmatic based on what you would see at the whole muscle and what would be anticipated would be happening, specifically with the power of these type 2 muscle fibers. And so we went back and compared it to the literature, and we're absolutely amazed to see normalized power data in these 90-year-old individuals that was comparable to that of a champion sprint runner who once had the world record in the 55-meter hurdles. Just absolutely outstanding, amazing to us. Furthermore, we actually, interestingly, and again, very paradoxically, saw an elevation in the normalized power of these type 2 muscle fibers with spinal cord injury, which was kind of had us scratching our head as well. And when we looked back, there was some data from some of the labs in Boston, from Dr. Fielding's lab, and the hypothesis had been put out that we know with aging we lose muscle fibers, and we particularly have loss of these type 2 muscle fibers. And so perhaps what we saw going on here was a phenomenon where as muscle fibers are lost, it's really only the best ones, or a survival of the fittest phenomenon. Only the best ones are really remaining. And then to basically complement that, if we have just a greatly reduced type 2 muscle fiber pool, even though we're not necessarily using these fast explosive muscle fibers as much with aging, when we are, we're relying on much less of them. And so perhaps this provides a stimulus to complement that already survival of the fittest phenomenon and further enhance the muscle fiber quality of these type 2 muscle fibers. So that was really the first work that I worked on during my time at Ball State. And then this study really presented a unique perspective to that, where once again, consistent with what we've seen previously, we saw an increase in normalized power of these type 2 muscle fibers in the old healthy compared to the young exercisers. And then what was also interesting is we saw that appeared to be a bit attenuated in the lifelong exercisers. And I think, although we can't say for sure, a pretty reasonable hypothesis for that is simply that these individuals were using their type 2 fibers with aerobic exercise. And even though we might not have seen that reflected in terms of the contractile mechanics of these fibers, perhaps the blunted improvement in normalized power of these type 2 muscle fibers just reflects the preservation of the type 2 muscle fiber number, that maybe we did have a greater number of type 2 muscle fibers in these lifelong exercisers. And because of that, we're not seeing the improvement that we would see with less active aging and then much less healthy aging. So that's really the picture that our hypothesis for that phenomenon. Scott, anything to add to that?
That was great summary, Greg. I just highlight the muscle mass in our older healthy non-exercisers was less than the exercisers. That we've got that documented in the literature. So it tends to fall in that paradigm. And I would just say, when we do on some of the work we've done where we've trained athletes, gone through different training blocks, base training, things like that, where they're putting in more volume, you tend to see this same phenomenon where the quality actually goes down, so to speak. It's a bit of an adaptation to training. So I think that's a little bit what we're seeing here, but I think honestly it's more complex with the aging and the layer that's going on. And as Greg alluded to, I think it's perhaps an idea. It'd be nice if we could figure out exactly how many fast fibers they have. Maybe overall their pool of fibers is being held onto a little bit better. And when you did look at the old healthy, they had some in the highest category way out there that the LLE just didn't in that functional range. So I don't know if that's at one level it could be one of these sort of compensatory things to help, right? I think that's possible, but they're also probably getting smaller. So perhaps that's it's on its way out, right? And it's just it's the last sort of phase for some of these fibers, because clearly they got less muscle, it's been remodeled, and things of that nature that are going on. Just a complex topic. So I guess we speculate based upon those data, but it is pretty clear now, and I think most people would agree. You get into muscle, it says quality goes down, but you have a lot of things other than muscle that are in those scans that are coming into those conclusions. You've got, and then it really comes down to the recruitment and the testing that's done with the muscle. Whereas with the single fiber prep, you've eliminated recruitment, you've eliminated, other connective tissues, infiltration of fat, those types of things. So when you just drill down on the muscle cell itself, it does look like the quality of these fibers, as almost titrates out. 70-year-olds is higher than younger people, the 80s higher than the 70s, the 90s. It just sort of stepwise up as it goes through the process. I'm pretty convinced, based upon the data, that a phenomenon appears to be more or less, a real phenomenon that's going on in the muscle.
Thanks